Okay then. So for those of you who don't know me, my name is Larry Reiter, and I'm a professor in the Department of Neurology here at UTHSC. And I actually run a Drosophila lab. I have the only Drosophila lab here. We recently started a seizure model that we have used to screen for anti-epileptics in a particular disorder called Dup15q syndrome. And so today, I want to talk to you about that, which was work we published last year. And also, in the second half of the talk, I'm going to talk to you about how we want to apply that directly to off-label studies of seizure suppression in these kids with Dup15q syndrome. So, all right, with that, I'm gonna jump into it. As long as my computer lets me jump into it. I'm gonna have to click it, looks like. All right, so I touched on this. My lab works in multiple different models. We have done a bit of human phenotype genotype correlation work in the past. And we also have stem cell models of human neurogenetic syndromes, which I'm not going to talk about today. And then we have these fly models, and I'm going to talk mostly about that today. But first, I'm going to talk about this disorder that we've been focused on. So what got me into Dup15q syndromes in general was that I had some experience with a disorder called Angelman syndrome. And this particular syndrome results from in most cases, from a deletion on chromosome 15. What became interesting to me, because I had an interest in studying syndromic autism, and I was interested in this particular locus because it turns out that a duplication of the locus results in a different disorder, and that disorder is Dup15q syndrome. So this kind of connection interested me, and I knew something about the genes in this region, so I started to look into Dup15q syndrome. Specifically, my lab has worked quite a bit on a gene called UBE3A, and I recently sort of went back through the literature and defined a few things about UBE3A expression that I think were somewhat overlooked. I'll talk to you about that in a second. So this is the locus, and you can see, I don't know if I have a pointer here, but I sort of do. I can do this, pen, laser pointer. So this locus has several genes in the duplicated region. There's sort of a minimal duplicated and deleted region between breakpoint 2 and breakpoint 3 here. And you can see that by these brackets. And within that region, there are genes that in neurons are expressed from one allele or the other allele. For instance, there are paternally expressed genes that would be MAGEL2 and SNRF-SNRP-N. And so my lab actually does work on some of these genes as well, because we work using dental pulp stem cell neurons. We work on a disorder called Prader-Willi syndrome, which I won't talk about today. But if you look in red, there's actually 2 genes in this box, but it turns out really just this gene, UBE3A, shows maternal specific expression in neurons. And that's been known for quite some time. And there are several mouse models that show that this is true. However, I think there was something that was a little bit overlooked, and it wasn't really until we did the fly work that we started to look back in the literature. But I'm going to talk to you a little bit about some of the published studies of these mouse models of Angelman syndrome that actually show that glial cells don't adhere to this allele-specific expression. So this paper shows that this antisense transcript, which is critical for the imprinting, is not expressed at all in glia. So in fact, you see sense UBE3A expression in the wild type animals, the animals with paternal deficiency, the animals with maternal deficiency, but not in the animals with maternal and paternal deficiency. So you see the transcript. So that was very interesting to us. But also, you know, there's actual experiments that showed that there's expression of the UBE3A protein in cells that are GFAP positive in the brains of these animals. So that was also further evidence that we should be looking at glia. And then a paper came out using human brain tissue that showed that indeed UBE3A is expressed in GFAP positive cells in the brain. Good. All right. So where does that land us? Well, at the time, you know, we decided that we could do a lot of manipulation of UBE3A expression in flies, and we would see if we could recapitulate the seizure phenotype that's seen in duplication 15q. So what a graduate student in my lab, Kevin Hope, did was he tried to make flies have seizures. And how do you do that? Okay, well, flies actually do have seizures that look very much like seizures in other animals and in humans. And this has been known for some time. If you watch them go through this process, there's a paralysis, initial seizure, paralysis stage, there's a tonic-clonic phase, and there's typically a recovery seizure, and then the animals recover. So, you know, we wanted to see if we could generate this phenotype, which in flies is often called a bang-sensitive phenotype. And that's because when you whack the flies on the counter, they can start to have these seizures. And there's many mutants that are known to do this. So the way that we do this manipulation in Drosophila is we use a system that's called the Gal4 UAS expression system. And it works like this. I'm going to give it to you in a nutshell, I hope. So we can put any cDNA that we wish into flies with an upstream activator sequence that is from yeast, actually. And when we do that and use a selectable marker, we end up with these flies that have various DNA transposons inserted with the cDNA of interest, but they can be in different locations in the genome. In this case, we're talking about UAS Drosophila UB3A, UAS human UB3A, UAS UB3A RNAi. So we can make all these lines. And then we have the advantage of crossing these lines to many Gal4 enhancer lines. And the way these lines were made, for the most part, although now they're being made slightly different ways, they used to be made by letting these P elements randomly find genomic enhancers that they land next to and express Gal4 protein. And then the Gal4 protein will be expressed in the brain or in the eye or in the wing or in any other tissue you could think of, even temporally. And also sometimes you can get lines that are expressing in individual neurons, for example. So if you cross these two lines, you end up making Gal4 protein that binds to the upstream activator sequence that drives your cDNA of interest. In this case, it'll be the cDNA being driven in glial cells. I'm going to show you some of that data. All right, so we've talked about it, but let's look at it. So I'm going to start this video if I can, if it lets me. There we go. All right, so this is a video of a fly that's expressing Drosophila UB3A in glial cells. And it's already been vortexed. So we vortex the flies for a few seconds. And if you do that to a wild type fly, the fly gets right back up. You can't even measure the time it's out. This fly is out cold, as you can see. However, they're going to have a seizure here in a second, and I'm going to speed it up for you. And what you'll see is uncontrolled wing movements. See, the fly is trying to fly, sort of, but it's really just having uncontrolled wing spasm. It's not really flying. And then it's going to knock out again. It's going to turn over. And eventually this fly will have a recovery seizure and will start to try to get up. We'll just speed that up. He's almost there. And there he goes. So what we measure in the lab is the time it takes for those flies to start climbing up the wall. And we consider that the end of their seizure recovery period. Okay, so I mentioned Kevin. So Kevin did a series of experiments. He expressed wild-type Drosophila UB3A. He expressed a C to A version of UB3A that can't ubiquitinate substrates. And then he expressed UB3A using ELAV in neurons. All of these other lines are up here. So those flies just didn't have seizures, but the flies expressing in all glial cells using repo-gal4 actually do have seizures and are out for the first 30 seconds and then slowly recover. And usually not all the flies will recover. He looked at flies that were extremely young, so between 0 and 2 days post-eclosure, 3 to 6 days post-eclosure, and 7 to 10 days post-eclosure. And what he found there was that actually the flies that are younger, that are 0 to 2 days, are somewhat resistant to seizures, and that's this purple line. And the flies that are 3 to 6 days have this 30-second window and then recover, like I showed you up here. And then if you actually age the flies out 7 to 10 days, the flies will have a seizure and not recover. Many will die. Some will just have a very long recovery time. This also worked with using human UB3A expressed in the fly. The seizures aren't quite as dramatic as with fly UB3A, but indeed the flies do have seizures, and like 40% of the animals are seizing, and in the 7 to 10 day period. So this window interested us because we knew that the flies were pretty much out cold. So we wanted to use that to our advantage and develop a screening method to screen through compounds that could suppress seizure activity in the model. And what we came up with was using tools around the lab. If we had an open tube, which was a 50 mil conical that we cut the bottom out of, and then we induced the seizures in these animals and then dumped them into this tube, we could then recover flies that climbed up the wall into this vial, because those flies were not seizing. And the flies that were seizing for that 30-second window would be down here at the bottom, and we would collect them in this plate. So we could separate the seizure and non-seizure animals, and that started us on this journey to screening for FDA approved and otherwise approved compounds using this model. So the first library we finished is a library called the Prestwick Chemical Library. There's 1,280 compounds. These compounds are chemically diverse, bioavailable. They've been approved by either the FDA or other approvals that they've had internationally. And only about 16% of them are drugs that are developed for nervous system disorders. Many are in other categories that we're not sure whether they're going to suppress seizure or not. And that's the idea. We didn't want to focus screen. We want to be able to repurpose some of these compounds to treat epilepsy. So after we screened the 1,280 compounds, we found something around 18 compounds in the primary screen that showed at least 15% suppression in the repo driving UB3A animals. And again, the way we did that is we separated the seizing and non-seizing flies, and then by looking at the genotypes of the animals, we were able to tell who was a repo driving DUB3A animal and who wasn't. Right? From those, we identified some compounds that were of interest immediately. And that's because all three of these compounds are related to the serotonin pathway. And in fact, you'll notice as well that, I'll put my laser pointer on. You'll notice as well that we hit levodopa. So we hit both serotonin and dopamine in the screen. And we hit some other compounds that we followed up on that were interesting as well. But the main focus and what I'm going to talk to you about is the serotonin pathway. So after we did the primary screen, we took all of those compounds on the table, and we had to find a way to verify that in a secondary screening that they were actually real hits. So we made slightly stronger criteria in this case. We wanted to look for a 50% decrease in recovery time in males and females, and a significance value of 0.01. So if you look at this table, these are all the compounds that passed. And again, you see levodopa, you see minoprene hydrochloride, you see serotonin, you see mirtazapine. So all of those compounds that I mentioned that affect serotonin signaling were indeed significant in our secondary screening as well. Okay, so let's dig a little bit into it. You know, one of the things that made sense to us was, okay, well, if dopamine and serotonin come up, what happens when we add both dopamine and serotonin to the flies? And what we see is we get further suppression of seizure, which on these graphs is going to be decreased recovery time. So these are flies expressing UB3A and glia in all these cases. This is water. This is with 5-HT alone, with L-DOPA, and with the combination of both in males and females. We looked at some genetic mutants to see if we could change the recovery time. And indeed, when we use mutants that are in the serotonin or dopamine synthesis pathways, so PALE, DDC, TRH, these are all related to synthesis of serotonin and dopamine in flies. You see that there's an increase in recovery time, even in a heterozygous mutant background. So we thought we were going in the right direction by seeing that. And in fact, we go the other way around when we use this dopamine transporter mutation, because now there's going to be more free dopamine. And in fact, the recovery time goes down when we use this background, DAT background. Okay, so we got somewhere. And what we actually did is we got really lucky, because the first drugs that we hit immediately gave us information about which receptor they were acting through. So it could have been any of these receptors, but in fact, mirtazapine, one of the hits, is a pretty good antagonist of 5-HT2A, 5-HT2C, and 5-HT3 in Drosophila. So it left 5-HT1A as a candidate for how this mechanism was suppressing seizures. So then we went into a list of agonists and antagonists, known agonists and antagonists for either 5-HT1A or 5-HT2 or all the 5-HT receptors. And we had very clear predictions, except for here, of whether we would see suppression or enhancement of the seizure phenotype. And in fact, it followed exactly what we expected. And we saw suppressors of seizure when we used 5-HT1A agonists, right? And we saw enhancers of seizure activity when we use 5-HT1A enhancers. And of course, when we inhibit all of the receptors, we see enhancement of the phenotype. So how does this look when you spell it out a little bit? So you can see that here we've got serotonin, mirtazapine, and these other compounds that are agonists of 5-HT1A, and we get nice suppression of the seizure phenotype, even at low concentrations in the food. Serotonin being the best suppressor, actually, of the seizure phenotype. And then we get these nice enhancements when we add compounds that are known antagonists of 5-HT1A. This also worked using human UB3A. So we expressed human UB3A in glial cells, the same methodology, but we didn't see quite as extreme of a seizure phenotype, but they did take at least 40 seconds to recover here. And then we could suppress significantly that recovery time by adding serotonin or levodopa. And we had the same effects with enhancement with these other compounds that are antagonists. So the nitty gritty of it was that we could actually use either 5-HT1A receptor mutations or 5-HT2A receptor mutations in the background to determine whether we're looking at 5-HT1A or 5-HT2A. And in fact, we looked in males and females, and when you put the repo driving Drosophila UB3A flies into a 5-HT1A background, you see an enhancement of the recovery time, really kind of confirming our suspicion that the activity of suppression is through 5-HT1A and not 5-HT2A, which didn't seem to have any effect at all. Okay, now this takes a little bit of explaining. So Kevin also did some work on a sodium potassium ATPase known as ATP alpha that we had been pursuing for a while as a UB3A potential substrate. So if you follow this Western blot, you'll see that when we express UB3A in glial cells, so repo driving UB3A, the levels of ATP alpha go down. When we express the C to A version that can't ubiquitinate, the levels are identical to wild type. And when we express human UB3A, we actually see an effect too on ATP alpha. So we kind of had the suspicion that that UB3A was ubiquitinating and degrading ATP alpha. And so that would imply that the ATP alpha was essential for the suppression. And we tested that using a specific compound, wabane, that is specific for ATP alpha. So if we inhibit the function of ATP alpha, we see an increase in recovery time. So all of this directed us towards a model that we developed. And the model, if I can get back to the clicker, oops, I have to change this back. All right. All right. Well, we can talk about this first. I forgot this slide was in here. Okay, so again, we're on the same theme here, ATP alpha. So if you overexpress ATP alpha and at the same time express UB3A, so now you're kind of recovering the ATP alpha levels. You also see a suppression in seizure. If you add 5-HT, you see a further suppression of the seizure phenotype. And in fact, if you overexpress ATP alpha, you add 5-HT and levodopa, you basically go down to 0. So you've now completely suppressed the seizure phenotype in these animals, pretty much. So it led us to this model, and the model involves ATP alpha in glial cells. So again, we're expressing UB3A in glial cells, and the normal role for ATP alpha is to maintain potassium balance at the synapse, and that's a critical role of glia. So ATP alpha is in the glial membrane, and if we degrade ATP alpha by overexpressing UB3A, we get less ATP alpha in the membrane, we get increased potassium in the synaptic space, and that causes increased discharges in the neurons. So how do we know any of this is going on? We actually did an experiment where we basically tagged the glial cells with TD tomato, so that just shows you which cells are glia. And then we overexpress UB3A, and you can see a significant decrease in the fluorescent green signal for this dye called APG-2. And this is actually a potassium chelating dye that fluoresces when it comes into contact with potassium. So we could see this significant decrease in potassium levels within the glial cells. So moving on, we knew that we got good suppression of the seizure phenotype with the addition of 5-HT and levodopa. So when we look now at that same experiment where we express UB3A in glial cells, we see a decrease overall in that potassium staining within the glia. And then when we add 5-HT and levodopa, we actually go up even a little beyond the normal levels of potassium in those glial cells. So what does any of that mean? So what we think is going on is that the activity of 5-HT1A, and we're not sure which dopamine receptor, the activity of these receptors in glia is acting through PKA to activate ATP alpha. So this part of the pathway is already known. We didn't do that work, but it was known that if you stimulate 5-HT1A, it can activate ATP alpha through PKA. So what we think is happening is if you free up levodopa, or you have more levodopa or more serotonin, you actually stimulate this pathway, which activates the remaining ATP alpha to normalize this potassium back to control levels, and you stop seeing the seizure phenotypes in the flies. So one more thing about this fly model that is pretty critical for the last couple slides in my talk. So first of all, we tested this PKA antagonist. I don't think we actually published this in the paper, but so we tested that model, and we did show that the antagonist H-89 made it impossible for 5-HT to suppress seizures in the model. So once we inhibit PKA, 5-HT is no longer able to suppress seizures. But more importantly for the remainder of my talk is that we tested some other drugs, and one of them in particular was an FDA-approved 5-HT1A agonist that's used for depression, that's called vortioxetine. And in fact, that drug was able to suppress seizures in our model pretty efficiently in both males and females. Okay, so to conclude the first part of my talk, I'm going to go over a couple things that we found. We screened a 1,280 compound library from Prestwick, and we found 18 primary hits and 8 secondary hits that we validated. 3 of the compounds from the screen regulate serotonin in some way, so that would be serotonin, mirtazapine, and minoprine H2Cl. The receptor types, just so you know, are conserved between humans and flies, and we found that 5-HT1A agonists suppress seizures and 5-HT1A antagonists enhance seizures in our glial driving Drosophila uv3a flies. And then by combining 5-HT and levodopa plus restoring ATP alpha levels, we can completely rescue the seizure phenotypes and restore the potassium balance in glia. We think through PKA, and I've shown you a little bit of data on that. Okay, so I may even, dare I say, open up for a few questions about that part of the talk, because I'm going to move on and talk a little bit about what we propose to do. If anybody has a question. It's silent out there in Zoom land. It's really weird to give a Zoom talk and like, I can't even see anybody because no videos are on. Can you guys even talk? All right. Okay, so I'll move on to the end of my talk here. Oh, there's Caitlin. All right. Hey, Caitlin. You have a question? Oh, okay. All right. So in the last part of this talk, I'm going to talk to you about something we're developing at the moment. And that is, we'd like to be able to move from our Drosophila model really directly into humans, because the compounds that we're screening and the mechanism that we're seeing is not only conserved in humans, but the drugs are already approved in humans. So I'm in the process of trying to put together a team to do this. And the reason that I'm trying to do that is because I've worked in this space before, and I have found some things previously in kids with duplication 15q, and I want to really use that and springboard into a study that actually suppresses epilepsy in dup 15q. So I talked to you a little bit about this region. I'm going to briefly go over some data that we generated quite some time ago now in kids with interstitial duplications of 15q. So between breakpoint 1 and 3, or between breakpoint 2 and 3, they have duplications. The kids that have the severe seizure problems, they typically have these isodicentric duplications, which are more copies of the region than the interstitial kids. But I'm going to show you why we're interested in looking at the IDA kids and what we've done with the interstitial kids in the past. So this is data from a paper we published now in 2013, so quite some time ago. And these are all kids that have interstitial duplication. The focus of the study was sort of on the autism phenotype, but we collected a lot of other data just because we didn't know what exactly the interstitial duplication phenotype looked like. And one of the things we found that has turned out to be really interesting is we found something on EEG that we thought was certainly a variant. And we didn't know if it contributed to the phenotype, but we found it in many of these kids. Some of them had paternal duplication, some had maternal duplication. And that sort of implies that UBE3A, which is maternally expressed in neurons, may be biallelically expressed in glia and contributing to the phenotype. And it's not a neuronal specific phenotype. So I'm going to talk to you just a little bit about some characteristics of this, EEG signature. And really that is that it kind of looks like something like a signal you would see in kids that were on benzodiazepines. However, none of these subjects were taking benzodiazepines. What's even more important was that we could see it in these kids, and there was a survey of other kids with 15q duplication who were on benzodiazepines, and from that seizure survey, we found that either those drugs didn't have an effect, or they sometimes made the epilepsy worse. So this was a large 400 family study asking about which medications kids had been on. And it looks like the benzos were not effective in treating seizure in DUB15Q. All right, so what does any of that mean? Well, a colleague of mine, Shafali Jest, has moved this variant into a high resolution EEG study, and she found some interesting things that I think are really relevant, and I'll tell you why when we get to that part. But basically, she was looking for this signature and found it, and it's an elevated beta activity throughout the record. But I like the way that she did this study, because you get these really nice pictures of this phenotype, or this variant phenotype, and I want to show you some of those pictures. So this is a kid undergoing the study. They wear these hairnets with all these tiny little electrodes. So this is not a clinical EEG. This is a really high resolution EEG. They have to do this for a study, and it's not really a typical thing that they do. And then the output looks like this. And this is really one of the things that I think is striking, is that this beta power activity, you see it really all over the brain here. And you don't see it in kids that are either ASD, and at least the ones they pick that have autism, or typically developing kids. So it seems to be really unique to DUB15Q. What was also striking to me, but there weren't enough subjects to really prove this, but I was really struck by the fact that kids that actually reported having epilepsy had less signal, or even no signal in this case, and kids that never reported having epilepsy had the signature. That was interesting to me, because I think that sort of indicates that there's this sort of buildup to the actual seizure events in the brain. And I mean, I can't prove that, but I'd like to use this marker now and be able to see, even in the DUB15 kids that don't have epilepsy yet, we could maybe see changes in this biomarker if we treat with 5-HT1A agonists. So what I've been thinking of, and what I'm actually starting to do with some clinicians across the state of Tennessee, is we're going to try and use some of the FDA-approved SSRIs and 5-HT1A agonists that are currently available, and we're going to look for a decrease in seizure activity in kids with isodicentric 15q, specifically in the kids right now who have pharmacoresistant epilepsy, who've been on a lot of drugs, but are still having seizures. We're going to measure a few things, and so seizure diaries are important, but we're also going to try and look at this EEG biomarker in these kids before and after treatment. And we'd like to have them wear a monitor that we found that might work out for EEG monitoring. So the drugs that we're really talking about at the moment, unfortunately, vortioxetine is going off patent. So the company that owns the patent's really not very interested in doing an off-label trial. I think it's a really good candidate, but there are other candidates. So mirtazapine is a sleep aid. It's already been shown to be somewhat effective in kids with Angelman syndrome. So this is a really good candidate for treating seizures in these kids. It's also a 5-HT receptor agonist. And there's a company that's called Biome Therapeutics that we're approaching. They have a 5-HT agonist that they're using for other things, but we think might be really useful to treat epilepsy in these Duke 15q kids. So this is a picture that's really too old, and my lab is logged on, so you guys know we need to make a new picture. And Caitlin, who popped in for a second, is a grad student in my lab. Kevin graduated and went to University of Utah, where he still works on Drosophila models. Jung-Soo and Tracy were involved in developing the epilepsy screen and have since moved on. And Dr. Bidisha Roy was essential to finishing the screen in flies. And with that, I will take any questions that anybody may have, and hopefully we can have a little discussion.